8 years ago, I had a dream. I decided to pursue that dream, and this is what happened.
Sorry about that. Well, welcome to Benito's explanations, okay? I'm going to explain how to get dressed, okay? Is that helpful to everybody? Okay, everybody?
Dolphins, like dolphins can swim. Though nothing will keep us together. We can beat them forever and ever. Oh, we can be heroes just for one day.
So this is my message to you, to follow your dreams, even if you are a complete muppet.
Hello everyone, yes, today is a very exciting day in the history of Benito's explanations. This is my 200th video ever, and as much as I'd love to be, celebrating, going crazy out on the town, whoa, lads, unfortunately, I've got to do a video on the immune system. But don't worry, because once my exams are over, I promise you something special, and that will include the famous 200th subscriber video special. Okay? Wah! Right, okay, but today we're gonna carry on with our parasite series, but we're gonna diverge away from it slightly. We're gonna take a little bit of a step back today, because our next two videos are going to be about how the immune system deals with parasitic infections, whether that be protozoans or helminths. Okay, so before we go into that, we need to have a bit of a refresher on what the immune system is and how it works. Okay, so we're going right down to basics. Okay, now the vertebrate and the invertebrate immune systems differ, but considering that pretty much all the parasites that we've talked about in this series are human parasites, we're going to focus in on the vertebrate immune system, right? Now, there are two jobs that our body has to do to protect us from all loads of infection, because all around us there's parasites and bacteria and viruses wanting to attack our bodies. So our bodies must be doing something right for me to still be here. Okay? So, first job is to prevent parasite infection from happening in the first place. Okay, so by creating physical barriers, secreting mucus, tears, enzymes, yeah, all loads of things, okay? And if they fail on their first job, then what they have to do, if a parasite does actually get inside you, you've got to remove or ameliorate that infection, okay? So, and if that doesn't work, you've got to make sure that if you're infected a second time, you can remove the infection more efficiently. Okay? So these are the things that the immune system needs to do. So I've talked about how our hosts have a load of behavioural, structural and physiological adaptations to prevent infection. Okay? A great example of a behavioural one is with malaria. Obviously, if you live in an area where malaria is prevalent, you're gonna, wear protection, when walking around, and hopefully there should be something which should be more common, but have mosquito nets in your homes. You could be less prevalent to infection by a particular parasite for ethnic reasons. Okay, so Jewish and Islamic populations don't eat pork. right? So therefore, they stand no chance of being infected by the poor tapeworm, Taenia solium. Okay, so they're all right for that. So for the rest of the video, we're going to be talking about what the body does once infection takes place, when the first job is broken. Okay, so somehow a parasite has managed to get into your body. What does the body do then? Now, vertebrates have an acquired and an innate immune system. Invertebrates, on the other hand, only have an innate immune system, but we're not going to care about that for the moment. So we're going to focus a little bit more on the acquired immunity of the vertebrate immune system, the one which develops over time. Okay? So the cells from the immune system are mostly produced in the bone marrow. They develop from stem cells, so undifferentiated cells in the bone marrow. And there are two main types. There are the B cells and the T cells, and we're going to go into them in a lot more detail. The T cells develop finally in the thymus glands, yeah, and the B cells develop in the spleen. Now, they're called B cells not because of bone marrow, B for bone marrow, because it's T cells for thymus, yeah, but it's actually called B cells thanks to the bursa of fabricus, which is the site where these cells are produced in birds. Okay, bursa of fabricus. Okay, so while we're on the subject, let's talk about B cells then. What do they do? Well, B cells produce antibodies or immunoglobulins. Okay, and these are proteins, and they're made up of 4 protein chains, and they're specific to a particular epitope. Now, what's an epitope? Well, an epitope is a part of an antigen, which we all know what an antigen is. That's any foreign body which the immune system will react to. Okay, so it's a specific part of that antigen which is just the right shape for the antibody to detect and bind to it, like a lock and key mechanism, if you like. So it's important to note that a single antigen can have many different epitopes which are specific to a particular antibody. Now, we normally shorten immunoglobulins to IG because we're just quite lazy like that, really. So then, something which is unique to immune cells takes place. We see somatic recombination of genes. And what's the product of this? Well, this recombination is random, and it means that each B cell has an immunoglobulin with a unique specificity. Now, the technical difference between an immunoglobulin and a B cell is that when the immunoglobulin is on the cell membrane, so it's on the surface of the cell, that's when it's called an immunoglobulin, or an IG. Okay? When it's released from the cell and into the serum, that's when it's technically called an antibody. But we don't need to worry about that. So, when this IG binds to an epitope, it stimulates the B cell to proliferate. And at this point, it produces plasma cells and memory cells. Now, the plasma cells produce more antibodies which go on and destroy the parasite. We're going to be looking at how it does that in a second. And then the memory cells are involved in immunological memory. So it means if there's a secondary infection, then the immune system will react much quicker to that. Now, there are 5 types of immunoglobulins, so let's go through them, shall we? What fun! Okay, so the first one is IgM. That's a pentameric molecule. It's usually produced first, funnily enough. There's IgG, which is probably the most abundant immunoglobulin there is. There's IgA, which is a dimer, and it's present in secretions and mucous membranes. There's IgE, which is involved in inflammatory responses, and then there's IgD, which we don't really know the function of, believe it or not. Right, so like I say, if an individual is infected a second time, then there's a much quicker response, and that forms the basis of vaccinations, right? That's what they do. They induce a primary immune response, so then if you do get the disease, for real, then the immune system will be able to deal with it a lot quicker, and hopefully you won't get, the horrible symptoms of whatever disease you're vaccinated against. Right? So, what do antibodies actually do? Well, they directly interfere with the action of an antigen, So this could be by preventing enzyme function and whatever. They also facilitate phagocytosis, which is a type of innate immunity, which I'm sure you all know about, right? So, big cell engulfs the pathogen, breaks it down, and that's the end of it. And they also attract cells which release cytotoxic molecules. Oh, how evil. And this is called ADCC, antibody-dependent cell-mediated cytotoxicity. Woo! Right? And they also activate complement molecules. And I'll tell you what they are in a second. So this whole business of the B cells and what they do, this is called the humoral response. Right? Now, I mentioned complement molecules. What do? So, complement molecules, God, they're nasty little devils. They create pores in the cell membranes of cells, which, as you can imagine, it causes a lot of problems, because if you've got a leaky cell, then you've got lots of vital, chemicals, nutrients, water, whatnot. Everything diffuses out, you've basically broken your cell. And they also attract inflammatory cells, okay, so causing more havoc. Now these complement molecules are always present in the circulation, but they're only activated by these antibodies, where they all come in and they cause what's called complement cascades. Sounds very dramatic, doesn't it? Now before we go on to talk about T cells, it's worth mentioning the major histocompatibility complex, the MHC. And this is a set of genes, it's a gene locus, which is involved at differentiating self from non-self. Now, if you think back to the evolutionary biology series, we talked a load about the MHC on how it can be used to decide mate choice in humans. Remember all that? The sweaty t-shirt experiment, smelling the t-shirts, and how it correlates to specific MHC loci? God, those are days, weren't they? This all links in, because the MHC is hypervariable, so each one of us has a very distinct MHC type. Now there are two types of molecule. There's class 1 molecules, which define self, and then there's class 2, which are present on some immunological cells, which we'll talk about later, such as antigen-presenting cells. Okay, now, when organ transplants are done, this MHC has to be typed, because you want to, the organ which you're transplanting into someone has to have a similar MHC to reduce any sort of immune response. It's got a whole load of different names. In humans, it's actually called the HLA, just to confuse things, right? So that's the human leukocyte antigen, right? Okay, T cells, what do? Now the B cells was humoral immunity. T cells is all about cell-mediated immunity. And there are three basic types of T cell. There's T helper cells, or TH cells. There's T cytotoxic cells, or TC cells. And then there's also T memory cells. So, what do they all do then? Okay, so the T helper cells, or the TH cells, help other parts of the immune system to act on a particular pathogen, okay? And they interact, or they recognise these MHC class 2 molecules, which can be on antigen-presenting cells. So this is when a cell engulfs a pathogen, takes all its antigens, and presents them on the surface of their cells to signal to other cells out there saying, look, I'm infected, come and solve this problem, okay? Great signalling mechanisms going on within our bodies. So when this TH cell recognises the MHC class 2 molecule on this antigen-presenting cell, it will proliferate, producing clones with exactly the same specificity. Amazing. Now, these T helper cells are defined by this CD4 receptor molecule. It's that which binds to the MHC class 2 molecule. Okay? Now, also what they do, this bound T helper cell can release cytokines, and these are molecules which promote B and T cell activity, so therefore amplifying this signal. Many also of these T helper cells will eventually develop into T memory cells, which, just like the memory cells of the B cells, provide immunological memory. Okay, so help when there's a secondary infection, right? Now, there are two types of T helper cells, subtle differences. So there's Th1 cells, so these are involved in cell-mediated immunity, mostly against microparasites, so the protozoans and probably the bacteria, the viruses, all that sort of stuff. Yeah? Whereas the Th2 cells, the T helper 2 cells, are involved in inflammatory responses. Okay, and also they help against worm infections. Okay, so these are something which will crop up in the later videos of the series. Now, I've written the hygiene hypothesis on the board. Now, what's the hygiene hypothesis? Now, the idea is that in developed countries where people aren't as exposed to, pathogens anymore due to, increased medical advances, in these environments there's greater Th2 responses. So that leads to greater inflammatory responses, so greater allergic reactions, okay, because you're more sensitive. This is suggesting that our cushy, medically advanced lifestyles that we live in today is actually making our immune systems more sensitive because we're not exposed to as much stuff as we're growing up. So as we get older, we're more likely to develop allergic responses to things. Okay, so T cytotoxic cells then, the TC cells, what do? Now, the T helper cells were defined by the surface marker CD4, right? These TC cells are defined by the CD8 surface marker, so not to be confused. And they release, just like the complement molecules actually, they release perforin, which creates pores in the plasma membrane, so it creates loads of holes, loads of lesions, so the cell dies. But remember, this is cell-mediated immunity, and the cells which are expressing these antigens on their surface, telling the T cell, come over here, I'm in trouble. Yeah, these are actually self cells, they're normal cells. So this is what we call antigen presentation. The TC cells are recognising the MHC class 1 molecules this time, which defines self, right? Okay, so they're all the main cells involved in acquired immunity. Just a little brief mention on macrophages here. They're involved in innate immunity, but they're real, cell engulfing machines. They're not very specific at all. They just go around munching everything by phagocytosis, in its path. Well, anything that's foreign anyway. Now, it's May time, a lot of you may be getting a bit sneezy, yeah, because of the amount of pollen in the air, because you're allergic, okay? And this is all to do with this hypersensitivity, and it's all down to, as you might have guessed, IgE, because as we talked about earlier, that's involved in the inflammatory response, okay? So, the antigen, so, a particular epitope on a pollen grain, for example, binds to the antibody. Then these antibodies will bind to mast cells, which release a load of histamine, which gives you that inflammatory response. We won't go into too many details about that. So that is basically it. That is the immune system. But just thought I'd end to see how everything links together. Okay, because it is actually quite neat. And I've drawn a little diagram here, which may seem a little bit complicated, but it all makes sense when you look at it. So right at the top, we have got our antigens. This is when you're infected with something for the first time. When that's introduced into the body, that will activate B cells down here, but also will be taken up by antigen presenting cells, those special cells of the immune system, which will activate T helper cells. Okay? Those B cells will go on to proliferate, produce plasma cells, which will go on to make antibodies, but also memory B cells involved in immunological memory. Okay? So, these antigen-presenting cells will attract T helper cells and T cytotoxic cells. Right? Now, something I didn't mention was that these cytotoxic cells can also develop into memory cells. So they'll eventually develop into memory cells. Alternatively, they can just be activated and turn into T cytotoxic cells, which will create all those pores in the cell membrane and kill that infected cell. Okay? But as I've said, T helper cells can also mature into T memory cells. Now, if you're infected a second time, then it's all a lot more simple, okay? Because there's already memory cells, memory B cells, memory T cells inside your body. Okay, so that'll activate the memory B cells, which will create more plasma cells and more antibodies, but also will activate more T cells. Phew, God, what a mind boggle. But really, not that bad. Hope you enjoyed this video. Next time we'll be going on to applying all of this stuff to see how specific parasites can handle our immune system, because we haven't really talked about many parasites today. So join me next time where we'll be talking about how protozoans, combat all of this. See ya.